All right, well, welcome everybody to our online open house for the Cancer Center at Sanford, Burnham, Prebys. It's my distinct pleasure to be here today as the moderator of our discussion about precision medicine. I'm Ruth Claire Black. I'm a community advisory board member for at least 5 years or so now. I'm a cancer survivor, and I'm proud to work with a small group of cancer survivors and family members who have supported others through cancer who make up the Community Advisory Board. And our mission is to sort of be the interface between the great work that our scientists do at Sanford Burnham and the community, right? So we run these webinars, which before COVID were in person at our wonderful campus up on the Torrey Pines Mesa, but now are online. So we have a great group of people who are logged in today. I'm very excited about this discussion. And for those of you that have joined the webinar, if you have any questions that you want to pose at any time during the discussion, the Q&A chat is open. You can pop them in there and I'll be monitoring that. And I think Ze'ev will be looking in there as well. So welcome. Everyone, I'm going to turn it over to Ze'ev, and he's going to do some brief introductions.
Thanks, Ruth. Wonderful to have you with us here today. We'll spend the next few minutes learning about precision medicine, and we are honored to have Eytan Ruppin from NCI, leading the data science at NCI, and one who innovate some of the great advances that we have in precision medicine. And I cannot find or think about anybody better than Eytan to explain us about precision medicine. So we'll have an open discussion. Would have prepared some questions that she's going to start the discussion. And if you have any questions from the audience, we'll be more than glad to entertain it as well. Here at the Cancer Center at the Burnham, Sanford Burnham Prebys is celebrating 40 years nowadays, together with the 50 years of the Cancer Act at NCI. Group of about 40 scientists that are doing cancer research, basic cancer research. We are NCI-Designated Basic Cancer Center. And we're very proud to do much of what we do in collaboration with translational experts that we have here on campus as part of the Prebys Cancer or the Drug Discovery Center. So without taking much more of time for introduction, Ruth, maybe you want to start by some of the ABC questions so we can get on road?
Sure. So I think probably everybody that's on the webinar today has probably seen the term precision medicine in passing. They might have heard that thrown around perhaps at their clinician's office or when talking about their own health care. But let's start by really digging into a little bit about, from your perspective as scientists, what is precision medicine?
Eytan, please. I can compliment.
Sure. So the basic idea of precision medicine, I would say its mission is to measure whatever we can in an individual patient in order to inform the best treatment to the patient that really matches his medical profile. So, in traditional medicine, you reach a certain diagnosis and then, basically based on the diagnosis, a group of patients with the same diagnosis will be treated in the same way, while precision medicine aims to go beyond that and treat individually and specifically, possibly in a different manner, patients according to their individual profiles, even when they share the same medical diagnosis. Hopefully that is believed to bring better outcomes.
All right, and so I think, precision medicine is this large category, right? But then I think within precision medicine, can you tell us a little bit about precision oncology and how precision oncology and cancer treatments might be changing?
Yeah, absolutely. So precision oncology is just the same thing, but to do it for cancer treatment, basically. So if you look at cancer treatments and until the last decade or so, most of them were based on chemotherapy and radiation. And there's of course many different types of chemotherapies and of radiation treatment. But basically the concept of these treatments has been that you want to target and kill replicating cells. And the idea was that tumor cells usually, of course, are much more proliferating and replicating than non-tumor cells in our body. And by having such agents, you will get a selective killing of the proliferating tumor cells. However, the limitation is that we have many non-cancer cells in our body that also proliferate and replicate as part of the normal functioning. And hence you get this toxicity and side effects which limit the efficacy and the dosage that you can give to chemotherapy. And its selective profile of killing isn't ideal. So on the background of that realization, there came a new generation of cancer drugs, which are called targeted therapies. And these are drugs that are designed to target the specific individual alterations that occur in cancer cells. So in contrast, in difference from chemotherapy that aims to target the replicative cell cycle process of the cell in different fashions, which is shared between cancer cells and normal cells, targeted therapies aim to target very specific vulnerabilities that ideally are only on the tumor cells, hence gains much more effectivity and lesser side effects. This sounds like wonderful, but of course has its own limitations and challenges that I'll be glad to explain later on. Just don't want to talk too much, right? My turn.
So maybe I can complement this by indicating that the notion of precision, whether it's medicine or oncology, has to do with the information we are able to gather from the patient biopsy of the patient tumor or from the sample that we're actually analyzing. And we consider that valuable information that is based on either the DNA or better, what Eytan is doing and many others trying to follow, is RNA. So the material that is translated to protein, which is much more accurate and more informative than just doing it on the basis of DNA. And this is the information that provide the base to perform fancy calculations that allow to come with predictions as per therapeutic approach and so on and so forth. Did I put it correct, Eytan? You have the opportunity to.
No, you put it wonderfully, and I would say even I understood, Ze'ev. Explanation was so clear. But I want to say, and to add on what Ze'ev said, that the fact that we are developing really precise targeted therapies is paradoxically, perhaps initially unexpected, a double-edged sword. Because as we are learning more and more with the development of what we call omics technologies, the methods that enable us to characterize the molecular landscapes of tumors from biopsies and so on, as Ze'ev has described, we are seeing that tumors are way more heterogeneous than we expected. So they have different clones with different molecular architectures, and that creates a huge challenge. So as we are learning more, we are aiming to advance, but we also learn more about the formidability of our opponent. And the issue of tumor heterogeneity is a major challenge now in cancer treatment. And cancer, paradoxically, targeted therapies usually target only specific clones, only subparts of the tumors, because they are so precise. And that creates a challenge because many times you get a wonderful initial response, and that is, of course, highly important and beneficial. But at times, regrettably, resistance emerges. And now you may understand perhaps why, because you are targeting specific clones of the tumor. So the next logical step has brought cancer researchers to think of the next two steps in developing cancer treatments. And we can, of course, discuss them later and so on of interest. One of them is the quest to find combinations of targeted therapies and immunotherapies and so on to cover and target as much as possible different clones that may exist in heterogeneous tumors. The other, which is a whole huge topic on its own, is probably you've heard of cancer immunotherapy. That is a whole new line of research which aims to say, okay, let's not aim to kill the tumor cells necessarily directly because they are heterogeneous and clonal, and that poses serious challenges. Let's develop ways to harness the immune system to do the work for us and kill the tumor cells irrespective of their heterogeneous nature and clonality. So I would say there was chemotherapy, then came targeted therapy, then came a realization of the challenges posed by heterogeneous tumors for targeted therapies, and that then boosted the search for effective combinations and for cancer immunotherapy.
I would like just to add or clarify one aspect, and a very important one that you have related to it, and that is tumor heterogeneity. So that has been a big event in our understanding of cancer or cancer makeup. We looked at tumors, or we considered tumors for many years to be one bulk, one mass. And with the better technology that became available to us, we realized that there is no one mass. It's actually many different tumors that reside within that mass. And the different lesions or tumors that are within that big tumor have different genetic characteristics. And therefore, they need to be looked at and treated not as one, but as many. And that raised a big challenge and big problem at all levels, including the level of how we treat such. So that's why when we went with monotherapies, we got resistance, as Eytan referred to, by the clones that are different from the big mass. It's enough that 1% or 0.1% or even less than that of the big mass will have different genetic characteristics that will evolve later on as resistance. And that's what we faced in the targeted therapy. Went to immunotherapy, which is a way by which we relieve the brakes from the immune system so that the brakes are there inherently to prevent the immune system from attacking us, allow it to attack the tumors that amazingly were able to escape that therapy and successfully were treated by immune therapy. But even immune therapy got its limitation, and there are resistance that we learn about for immune therapy. That's where the combination comes about. And I think that understanding the complexity is one of the challenges that we scientists have all the time try to run the battle one step forward in order to see how we can overcome and be smarter, at least for a short time period until we figure out something better. Did I put it right?
Absolutely.
So this sounds very different than the kind of cancer treatment that I went through, and maybe a lot of people who are on our webinar today who've had some cancer treatments themselves or who've known some people who've gone through it. You know, I think even today in 2020, 2021, there's a lot of this standard treatment protocol that's rolled out by your oncologist, very much a one-size-fits-all approach. And it sounds like precision medicine and precision oncology is moving us in a much more individualized direction. So can you talk a little bit about how these big treatment protocols today might be different in the future under precision medicine and precision oncology?
Yeah, so first of all, to say I want to first of all acknowledge my limitations. So I'm an MD, PhD in my training, but I'm not an active oncologist. I'm actually a psychiatrist in my residency, so not very relevant to what I do or to our discussion. So I'm not treating patients on a day-to-day level, but of course I work with many, I'm very fortunate to work with many leading physicians at the NIH Clinical Center, where we treat, more advanced tumor patients and more aggressive and so on. So we do a lot of different efforts on precision oncology in the NIH and the NCI Clinical Center. But I totally acknowledge that it doesn't reflect the standard oncology practice in many places in the US, in the world, and even in the US. The issue is that it takes a village. These are sophisticated approaches. It takes time. And there are also inherent limitations. So with current standard, what we call mutation-based, fusion-based targeted therapies, we estimate that currently with the way we measure the tumor molecular status, which are called different mutation panels, if you're aware of that, we can offer solutions and help about 10 or 15% of the patients. So with current targeted therapy approaches, the majority of the patients, even in leading medical centers, cannot benefit from them. This is, however, gradually, and I would say rapidly changing in terms of patients' eligibility to immunotherapy, certainly for second-line treatments, but even more and more for first-line treatments. And I have to say, in my mind, to the credit of the FDA, it has been quite aggressive and fast in authorizing more and more immunotherapy and more cancer patient indications. And I would say that in specific cancer types, I can say which they are, currently a large fraction of patients are eligible, both by FDA and insurers, for different checkpoint therapies, what we call I should comment that immunotherapy also comes in different varieties, and if you want, I can explain a bit more on that. But gradually, immunotherapy is taking a larger place in the current practice of patients in the US in quite a few leading tumor types.
Well, this is exciting. And we do have a great question here. Somebody asked, are there better success rates with certain types of cancers using precision medicine or precision methods versus the standard treatments? I think that's a great question.
Oh, I'll take that question. The answer is obviously yes, because otherwise they would not have been approved by the FDA. You see, the way these treatments or any cancer treatments are getting approved, they have to show a significant improvement over the current SOC, the current standard of care. And many times it is done by what we call double-blinded, multi-arm clinical trials, where a same cohort of patients is randomly assigned either to treatments with the standard of care or with treatments that with the new drug, which was on a clinical trial to be approved, and you have to show a robust and significant improvement. Okay, otherwise you will not get approved. And a clinical trial is designed to take some time in a double-blind manner for a certain period. Usually it has 3 outcomes. Okay, one of them is that what we hope most, that the drug is so successful that the clinical trial is stopped in the middle because there's no point to continue, because it shows so much evidence that you want to treat everyone on the trial with the drug. The negative outcome is when you do the whole trial and it doesn't show efficacy, or there is a lot of toxicity. And in other times, the trial is concluded and you do show efficacy. The drug may get approved, but it wasn't so stark that, you know, it was stopped in the middle.
All right, we had a couple of other great questions here as well. So a good question here is saying, these precision medicine and precision oncology approaches that you've spoken about, do they rely or do they sit on top of current existing chemotherapy drugs and treatments, or are they different from that?
Right, so they are conceptually and fundamentally different. And I explained a bit, I can explain more. But if you are asking whether in practice they are given in combination, the answer is, it depends. Sometimes depends on the clinical context, the cancer type, the clinician appraisal of the patient's status, the aggressiveness of the tumor. There's many clinical considerations, and the clinician may sometimes decide to give them either as individual regimens or in combinations with, as you said, existing chemotherapy and radiotherapy and so on and so on. And we are actually learning a lot more on how to do these combinations in a biologically motivated, sensible manner. And if it's maybe of interest later, I can explain more about that. I hope it answers the question.
It does, yes.
A couple of questions relate to hematological malignancies. And perhaps, Eytan, you can address how precision oncology can also address hematological malignancies in a way we do it today.
I will. So thank you. Thank you for these questions. The story of hematological malignancies is indeed very different from solid tumors. And actually has a few interesting lessons which I can reflect upon. First of all, I don't know how many people in the audience are aware, but one of the tremendous advances that have been made in cancer treatment over the last, I would say, two decades and so on, has been in pediatric hematologic malignancies. It was a tremendous killer, and it's just an amazing success story where currently a lot of previously, you know, disorders that kids would die, now they're cured. Okay, it's really, and that's a subject, you know, we can talk for hours just on that to go, but just keep it in mind. It's a huge success story. I wish we would have such levels of success in other cancers. We don't, okay? Pediatric hematology is a huge success story. However, again, you see the foe is very serious, and I'm not in the business of sugarcoating things. I want to give you the realistic picture and the challenges. We do now see later on with patients, pediatric patients that were cured, we do now see a phenomenon of secondary cancers. Cancers that we think, we have evidence, may have been caused by the therapies which cured the kids, but later, 20 years, 25 years, and so on, we see recurrences of different types of cancer, not necessarily hematological ones, which we think are related to the treatment.
Yes, we've had some great questions on this very topic, actually, while you guys were having this discussion about recurrence and resistance and how precision medicine is working to sort of address and overcome those issues.
Okay. With your permission, Ruth, I want to finish to answer the previous
Absolutely, go for it.
Because it was asking about hematological malignancies, and I just mentioned the success in pediatric malignancies. I want to mention two additional aspects. One of them is that another type of hematological malignancy, CML, chronic myeloid leukemia, is another huge success story. This is in adults of targeted therapy. So lo and behold, this is one of the greatest successes of target therapies. It's initiated by a specific fusion event in a gene. I'll spare you the details, but cancer researchers have managed really ingeniously to develop a treatment that targets that driving fusion, and this transformed the prognosis of this disease, CML. In general, fusion events are wonderful opportunities for treatments. We need to invest more in sequencing tumors, in collecting data, in making it public for everyone in order to identify more such treatment opportunities in other cancer types. Quite a few of the recent approvals of targeted therapies are targeting fusion events. It's a wonderful treatment opportunity. They are relatively rare, but still we are not doing enough, okay? So we are making advances also in adult hematologic cancers. Lastly, big strides in adult hematologic cancers are actually done with immunotherapy, with a specific type of immunotherapy that you may have heard of, perhaps, CAR therapy. So, and I'm proud to say, a local patriotism, a lot of this research has been developed originally in Israel. I'm an Israeli, I admit to the fault. In the Weizmann Institute, and later on in my home institution in the NCI by Steven Rosenberg, who is one of the NCI's leading candidates for a Nobel Prize in Medicine, hopefully for these developments. So what you do, you engineer immune cells, a specific type, which is called T cells, to attack specifically the malignant cells in ALL, in AML, and this is a very promising treatment and research direction. There's, of course, a lot of work to be done, but there's already companies worth of billions of dollars in this space developing and generating such treatments. They're very successful and helpful in hematological cancers. They're still not making the change that we want in solid tumors. This is an ongoing challenge, and we are beating our heads, including here in the NCI, including efforts in my branch to try and help NCI researchers in that aspect. It's still an ongoing open challenge.
Well, I'm delighted to hear you talk about the CAR cell therapy, because we had some great questions about that. You know, people who have heard about it were asking about the pros and cons as part of precision medicine, where it fits in. So I'm really delighted to hear you make those comments.
So you want me to address that question, or the pros?
Yes, if you have time, that would be great.
Sure, unless Ze'ev wants to pitch in, because I am talking like there's no tomorrow, and I usually prefer to listen.
No, I think that the CAR-T is an interesting direction that we are fully aware of, and Eytan, you can relate to that. But I want to go about resistance, the topic that came again and again, and one of the biggest problems we have in clinical oncology, from a different perspective, and maybe escalate or upgrade what Eytan is talking about to some of the technology that he developed over the last few years, which has to do with synthetic lethality. And I'll give you an example of a study that we've conducted where the technology that Eytan developed helped solve a very serious problem, and that's a problem of resistance. So many years ago, probably about 20 years ago, there was a drug that got into the market, FDA approved, and successfully used to treat ALL, one of the leukemia types. The same drug was approached or tried on some solid tumors, pancreatic cancer, breast cancer, and failed miserably. And the question of why it does work in leukemias and fail in solid cancer has been open for many years. And we have approached this question and tried to understand what different is between the solid tumors and the leukemias. And found that there is a pathway that is activated in the solid tumor. There is one signaling event that makes or conferred the resistance. And that's why solid tumors don't care about the drug. With that, we approached Eytan and told him, you know, we need to find a partner drug for this in order to overcome this resistance. Can we do that? And thanks to the computational approach that Eytan and his folks have developed, we were granted a list of about 5-6 genes that, based on prediction, would allow to overcome resistance. We tried, and they worked. And that led us to come up with a combination therapy, where one drug was already there, but failed on its own to do much. And the second drug, which was predicted by the computational approach, made the first drug work remarkably well in pancreatic cancer and melanoma. And this is a study that, although published a couple of years ago, is headed toward clinical trial in patients over the next few months, I believe, thanks to this computational ability. And I call it the precision medicine that started in the lab, started in a given set of samples that we have analyzed ourselves, and was able to be taken further up to the clinic. So when I said synthetic lethality calculation, I refer to a combination of two drugs, one of which in each case is not sufficient. And Eytan, maybe you can refer to explain a little bit the technology that is beyond that allow this combination to work so. And that's not a simple task to explain.
I can do that, of course, but I'm not sure we want to. I'll let Ruth, steer the discussion, perhaps, because I have respect to our audience, and I'm not sure they will like this suggestion. But Ruth, whatever.
Okay, well, lots of great questions coming in. And so I think two areas where people are interested in hearing a little bit more. So people want to know if there are other exciting success stories or other positive aspects of precision oncology that they should know about or that you want to share. And then sort of on the other side of that, I think people sort of are interested in knowing on the other side of the success stories, are there any significant limitations or challenges that they should know about or that really sort of damper your enthusiasm for where things are going?
Yeah, wow, what questions, what questions. There are quite a few additional success stories of targeted and immunotherapy. I do not want to take the time to enumerate all of them, but just to mention a few. So targeted therapy has shown considerable success in a few cancer types where you can identify very specific molecular alterations for which the drugs can target and are effective. As I said, many times after initial success, resistance can emerge, but it's not always. And sometimes you do get, quite long-time responses in terms of years and quite a few years and so on. And that's, of course, progression-free, what we call progression-free survival and with a reasonable quality of life. That's, of course, huge when you can do that. So that's extremely important. Typical lung cancers, typically in a specific type of non-small cell lung cancers, in renal cancer, in melanoma, and a few other cancer types. Immunotherapy is certainly, and I think that's the reason why there is such an excitement in the cancer research treatment community about that, is the first time, since surgery or, complete elimination of early tumors, where we can see durable responses. And for a considerable fraction of patients, and in a few cancer types, in melanoma, in lung cancer, in kidney cancer, in bladder cancer, beginning to show value in triple negative breast cancer, which is an aggressive form, and gradually more and more. There's a huge effort invested in this direction, guys. I can assure you that no effort is spared. Just to give you an idea, there are thousands, literally thousands of clinical trials ongoing on as we speak, trying different things. Some of them are not rational based, some of them is just trying, but okay. And just let you know, just in case if you're not aware, I would like to mention that there is a wonderful site called clinicaltrial.gov, in which any person, any family member, any patient can go on, describe some characteristics of a disorder that interests him, and get immediate information on all clinically relevant trials, where they are occurring, are there queuing patients, any information that is available on them, any prelim results. And that's an amazing resource that I think maybe some people may not know about it. It's called clinicaltrials.gov. Okay, so all information on all clinical trials is publicly available to everyone. So I answered just the first part of the question. You also asked me about the challenges and where I think the money is and where it's going ahead. I'll be happy to answer that, but perhaps Ze'ev wants to comment, or you want to lead the discussion elsewhere, please.
No, I think that this is a very important point. What are the difficulties we now encounter, and how we may overcome it? And we touched upon heterogeneity and resistance, some of them, but how you envision going about them is going to be very good. Another question that comes about by some of our attendees has to do with, oh, this is great, but where can I use it? What can I do in order to use it? So that's another thing that we need to somehow reach and address. But take it from the first, Eytan.
So the challenges that we face are formidable, and in two words, to echo what we previously said, it's a tumor heterogeneity which causes resistance. The way to move ahead involves a few components, I think. And I'll tell you my thoughts, but of course, it's just my thoughts. Others may think differently. It's a huge space. I'll tell you my thoughts. My thoughts is that first we need to do additional basic science to understand the molecular underpinnings of the tumors that we see. We do not know enough. We need to collect more data, analyze it, and better understand. What helps us now do it are tremendous technological innovations in our ability to measure what's happening in the tumors in a single cell resolution. These are things that are happening as we speak, just in the last 5 years. The pace of these technological developments, of our ability to see, to measure, to understand what's going in the tumor are revolutionary. And I'm not saying that lightly. I think that on one hand, it floods us with data, and it's a tremendous challenge to data science like me. On the other hand, we will gradually learn how to harness this data and find selective, effective combinations with minimal toxicity, which are tailored for patients. We are, my lab, and I'm sure many other labs in the world are trying to develop ways to do that as we speak, guys. To what level we will succeed and when, I cannot say. I am sorry. I just can tell you that we are trying our poor best. I want to highlight another tremendously promising direction, Tumor. One of them is, in addition to the technologies that we are developing to understand and measure each individual tumor in a patient, we are developing new technologies to deliver drugs selectively as much as possible only to the tumor cells and not to other tissues and organs. So, you know, currently, basically, we are giving drugs to patients either per os or IV and so on, and they reach, most of the times, they reach most of the tissues in our body, and hence the difficulties. But there are new technologies, just to name a few. You may have heard of nanoparticle conjugated antibodies, degraders, and so on, that hopefully soon will allow us to deliver the drugs only to the tumor cells as much as possible. And that will be transformative. And finally, an exciting, amazingly exciting new development in technology is that of liquid biopsies, what we call. The ability to measure and detect very small fragments of tumor molecular material in the blood. And that has two major benefits, ramifications. One of them is to monitor the success of our treatments in an easy, continuous, real-time level, and actually see if you're giving a patient a treatment, if it works or not. And the other, not less exciting, and which would have tremendous impact on cancer morbidity and mortality and so on, is early detection. The ability to have a test that you can perform, you know, annually or to populations and risk and so on, and detect cancer very early. If you look on plots of cancer survival and success of treatment, you will see that a major factor determining the outcome and the prognosis is the early detection. Most cancers, even very aggressive ones, if early detected, are curable.
So while you've been talking, we've been getting some great questions kind of in that area, right? So people picking up on that there's a strong connection here between genetic research, possibly doing some genetic testing and precision medicine. So, you know, people wanting to know, you know, should they be doing genetic testing? Should they be kind of monitoring themselves genetically? Would this help or hurt any implementation of a precision medicine program?
Sure. Ze'ev, you want to address that?
Go ahead. I think that goes very well with the early detection part that you just ended with.
So we should, in response to this question, we should distinguish between two scenarios. One of them is the standard genetic testing, which, you know, companies like 23andMe and others and so on, you know, you spit into a tube and you get an assessment of your genome and so on and so on. The ability of these technologies to inform you about cancer risk via what we call GWAS, genome-wide association studies, is very limited. Okay, unless in very specific cases For example, you may have heard about germline mutations in BRCA1 or 2 in the case of breast cancer, ovarian cancer, and so on and so on and so on. But by and large, the predictive power of these genomic tests for cancer is quite limited. What I am talking about is a different technology, which is just, being developed as we speak. And that is monitoring the blood, looking for specific molecular signatures of presence of tumors in our body. This technology is rapidly developing, okay? I hope and believe that it will be a game changer, as I said, both in early detection and in our ability to monitor the success of treatment in real time, which on its own is extremely important, because if you get early indications that the treatment doesn't work, then the oncologist, can switch to something else. He can think again. Currently, the oncologists are limited.
So we're getting some great questions about this coming up in the chat. So I think there was a lot of excitement around some of these game changers around using the immune system to actually kill the cancer cells, right? So people were asking, what should they be doing to improve their immune systems? Are there any ways to improve the immune systems to help with this kind of an option?
I would say, so there's a lot of studies, and Ze'ev actually is a world expert on this topic, on the relations between diet, microbiome, and immune activity. So Ze'ev is one of the few top people in the world to answer there. Ze'ev may not like my answer, but I think that these effects are marginal. This is my hunch. What happens in tumors is that the tumors actively change their environment in order to suppress the immune activity. So really you need to intervene actively and release the brakes that the tumor imposes on the environment in order to reactivate the immune system. So that's my take. Ze'ev, perhaps you want to have a different perspective.
I think that as long as we are talking about early detection, that is before tumor emerge, or when tumor emerges very, very small, then the likelihood of managing to control the situation and provide a specific therapy is very good, and the likelihood of success is very high. But in most cases, and this is where we should aim at, we have the technology, we should be able to do so. But in most cases today, this is not where we are. We are dealing with tumors that are much bigger. They already emerged. There are metastases already in some cases. And the problem is much more complex. And that's where the resistant heterogeneity come about, because we're no longer treating a given lesion or a given mass. We're treating many, many different masses within one tumor. And that's where the technological advances that we are making, thanks to computational know-how, allow us to use better tools, and immunotherapy is one of them. I think that in all cases, one answer is very clear to us. A single bullet is not going to make it. That we are not going to have a single bullet for any cancer. And when we speak about combination, then we need to consider all options. And immunotherapy combination with targeted therapy is one. Immunotherapy together with chemotherapy is another, and so on and so forth. And we need to be aware of what is that at hand, to best predict or calculate what would make best sense logically to go in terms of therapeutic approach. One thing that I really would like to emphasize, we spoke about it early a little bit, is that notion that we focus on RNA, which is a specific material that we extract from the cell, as opposed to DNA. And I have to emphasize it because for many, many years, we have held precision or personal medicine based on DNA assessment. And Eytan mentioned BRCA1, 2. There are about 300 genes that are known to be mutated or screwed up in cancer. And for years, we have followed them up and tried to use that information in order to predict possible therapy. This did not go well. This did not succeed on its own. And we know that today. And that's why we have to use technology that rely on greater resolution. So imagine you look at the map of Washington, D.C. or Los Angeles, and you look at a very high resolution, not detailed map, and you see one blob of roads. You go to high resolution, you see many, many roads going all around. And this is the resolution that we are able to get with RNA technology. This is the material that we can use to compute what might be the best therapy for that given sample, because that reflects what are the activity that happens in this given tumor at any given time. And that's why we are making the progress that Eytan has referred to in his discussion. I hope that was clear.
Well, we're working towards the end of our hour and we won't go over in time. So I just think now you guys have told us a lot of great things that's going on in the science, the hard work that's happening on the science side, but let's turn this around for a moment. What can the community, what can we as the community members, what can those of us that are on this webinar today, what can we do to help move precision oncology and precision medicine forward? Are there any avenues that we should be exploring that would help the scientists?
Yeah, I think so. I think so. I can point to a few. I'll mention two of them, and then perhaps Ze'ev can add. One of them is that the sorry state of affairs, I can tell you as chief of data science in the NCI and CCR, is that there is a lot of data out there which is extremely valuable and is not publicly available to all cancer researchers in the world. A lot of this data is even funded by your money, okay? Public money, taxpayers' money in the US and so on. I failed to comprehend that. I failed to comprehend this state of affairs. I am trying to do the little I can within the NCI, the NIH, to change that. It's a complex issue, and I don't want to simplify it. It's not black and white, and there are different sides to the coin. But I really think that we should move forward and make at least all data that is publicly funded available to all researchers, because that's our hope, you know, to move forward. When data is only shared in the eyes of a few, so much is wasted. Furthermore, there's a lot of data which is generated by the pharma, okay? Not directly from our money, from the taxpayers' money, but from revenues, right? And I think, while I understand the IP issues and proprietary issues and so on, I think that more thought and effort should be placed in having a process where this data is gradually, after some time, released to the public. And I think that as a community, you should talk to your congressmen, your senators, and urge them to take action and devise a new regulatory system that will entice or force pharma to make their data available. Beyond that, so that's on the data side. On the treatment side, there are issues. There is a chicken and an egg. There is a certain vicious cycle that needs to be broken. You see, in targeted therapy and in immunotherapy, in order to develop new treatments, you need to measure more things. Measuring more things costs money. Now, the health insurance don't necessarily want to pay for that. And they say that they don't want to pay for that because currently there aren't treatments that are approved based on this data. But if this data is not collected from patients and we can't learn from, how can treatments be approved? So there is a vicious cycle that again is non-trivial and deserves more thought. Now, I am confident that in 5 years or 10 years, the health insurers will approve these things. It's unstoppable. But I urge to do it sooner than later.
I would just add to that, very well said. I would add to that the patient perspective, and will emphasize it a little better, and that is that if I need to, I have a biopsy that is to be analyzed by a pathologist, I'm in a position today that can request my oncologist to have it analyzed also by molecular tools that will provide the data that the computational approach can use to predict much greater resolution predictions than otherwise, or what we have been doing until now. And that's one thing that we as community, as patients, can approach and get the information that can be used for prediction in a better resolution.
Well, that's been a great hour. Just to let you know how appreciative the participants have been. There's lots of thanks coming in to all for a great webinar. People saying it was very informative. They learned a lot. And just kind of to close out, some people are asking about where they can learn more. So asking about what's the best way for non-scientists or community members to educate themselves a little bit more about precision oncology, immunotherapy, and some of the wonderful things that you've talked about today. And then we're going to leave it there for the hour.
Wow. This is one excellent question that I do not have a good answer because, you know, I'm immersed in that over the years and I'm talking to people following the literature. I'm no more an innocent bystander. I'm not very familiar of, because things are developing so fast, of any, you know, off-the-shelf wonderful book, you know, but I'm sure people can search for it. I can just comment anecdotally, if you haven't seen, there is this wonderful movie about James Allison and the miracle of developing and inventing immunotherapy, which of course I don't remember the name of the movie. Ruth, do you know by any?
I don't.
No, so look, excuse me?
Breakthrough or breakout? Breakthrough.
Breakthrough. So it's a wonderful start, and it's an amazing story because immunotherapy almost didn't happen. We are very lucky that we have immunotherapy, and if you want, I can explain. But so I think since the pace of things is happening so fast, you have to search in the internet. I at least am not aware of very good, you know, textbook-like and so on and so on. The internet today, as you know, is an amazing resource, but it can also be a bit dangerous and risky. I don't have good solutions. Ze'ev, maybe.
Well, I'm just going to say, I'm going to jump in here and leave it there and say that that's, I think, what we're trying to do here at the Community Advisory Board for the Cancer Center. We're trying to make these kind of difficult science-based topics accessible to the community by inviting scientists such as yourself to answer our questions and talk with us for an hour. So for everybody else that's out there on the webinar today, keep your eyes peeled for the next Sanford Burnham online webinar. We'll definitely invite you back to join us again. We hope that you will. We've got some really exciting topics lined up for the rest of the year. And to keep it at the hour, we're at the hour exactly. I'm going to sign off and say thank you to Ze'ev and Eytan for this wonderful hour. And I have learned a great deal. Thank you.
Thank you so much.
All right.
Thank you, everyone.
Thanks.
Bye.